Hello, my name is Luciano Costa. I'm a myeloma physician at University of Alabama at Birmingham. And I'm here in the International Myeloma Society annual meeting in Toronto, Canada, where I had a chance to moderate a symposium addressing some innovations with CAR T cell therapy, but also the potential for enhancing the immune therapies and ultimately anti-myeloma therapy with the new protein degraders. So let's talk about that first part a little bit. I'm sure most of you either have had personal experience or have read about CAR T cell treatment. Of course, is a revolutionary treatment that has caused a lot of hope and a lot of enthusiasm and some outstanding success. But the CAR Ts that we have now are really only the first generation of that. They all target BCMA, this protein that I'm sure you heard about, B-cell maturation antigen. And they have a lot of opportunities to improve on both efficacy and safety. So a lot of what we discussed was about how much we have learned from the real world, from treating real patients outside clinical trials that have, characteristics that are not necessarily the same. People with impaired renal function, people with poor performance status, people who are older. And it's interesting to see that can be done for the most part very safely. So we're extending the benefit of existing CAR T cell therapy to more and more patients. But the other part is what is in the future, right? How can we overcome some of the barriers for CAR T cell? And some of the things that are very promising are CAR Ts that go for a different target, such as GPRC5D, and that can be used as a second CAR T or a CAR T before a BCMA CAR T, or quite interestingly, we are researching the use of a CAR T that targets both BCMA and GPRC5D at the same time. And that's, of course, incredibly promising, exciting. Another target that might come up is CD19. There's at least one CAR T cell in development that is very active against BCMA and CD19 at the same time, which might have some therapeutic advantage as CD19 is expressed in precursors of multiple myeloma, like in the B cell before it evolves into mature plasma cell antibody producing. The other possibilities are using CAR Ts that come from a donor that bypasses the need to wait 4 to 6 weeks for manufacturing and can allow people to have immediate access to CAR T cell. And perhaps in a little bit more distant future, the in vivo CAR T. So instead of collecting cells from the patient or from the donor, modifying those cells, we actually give the virus straight to the person. The virus goes to the T cell and produces the receptor, and that T cell becomes CAR T. So that gives people immediate access to that therapy potentially.